Hello, I'm Ed Schulak. Over the last 14 years, we've built a whole conglomerate of companies focused around aging and the holy grail of aging. We have really focused this area in genetics and gone off on many other different pathways from that lead. In 2010, I attended a conference out in San Francisco, which theme was longevity. And to tell you how this field has changed, this group looks a lot more sophisticated than the group that I saw out there. They had people with top hats that looked like chimney sweeps. It was a big crowd, maybe 300 people. I'd say probably a third of them doctors. And the conference was mostly at that point in time, just 16 years ago, based on telomeres. That was really the field. That was what everyone thought would solve aging. And they said, here are these little plastic caps at the end of the shoelace of our genes. And we noticed that they keep declining every time a chromosome makes a copy. And if we could but keep those steady, then we would know that we could solve the aging. And as you know, life went on and we started working with telomerase. And telomerase had the one defining characteristic of keeping cancer alive. And so that didn't work so well. But curiously, at the very end, the very last talk, which will be embedded in my head forever, was last chance. What you do if everything else fails. And the guy with the top hat gets up, goes to the front of the room, and says, here's what we're going to do. He says, if none of these treatments work, he says, we have a cryogenic company, and we're going to freeze your body like Walt Disney. And if you can't afford the whole body, we'll just take your head. And this will forever be indulged in my memory as the field of aging. Fortunately, I think the last 16 years has been incredibly productive. I chose as my theme the Edison Lab, and I've always been inspired by Thomas Edison and what this small group of 100 people created in such a short period of time between around 1880 and 1930. And this was really, if you remember, the dawn of electricity. They had an entirely new tool to play with. And everyone knows of Edison's creation of the light bulb. Not many other people know that he also made a phonograph. He made the first electric motor. And this was such an incredible period of time, over 1,000 patents, which, truly built the modern revolution of industrialization takes command. I've tried doing a similar thing, maybe not quite as successfully, but we've certainly had a great time, and it certainly led to many places that nobody expected. So it started with a journey in 2011. I decided I'd go meet and live with the top 10 living geneticists. And if you recall, this was the beginning of modern genetics. It was only in 2001, 2004, that we actually had a reference gene, which held about 80% of the human chromosome. And everyone thought at that moment in time, that was it. We were going to solve all disease. They found 23,000 genes. They were convinced that one gene was responsible for each disease. And life turned out to be much, much more complicated. As we now know, very, very few genes are directly related to a single disease. Most of them are multi-complex structures. We built within that year a company called Dovetail Genomics, ARC Genomics. And these were companies that were working on even more precise ways to look at the genetic code. And these companies are very much vibrant today. And they have really laid out a future of a highly detailed genetic code. It's still the most perfect genetic code if you wanted to understand a whale. You would hand Dovetail a little piece of the tissue. Dovetail would hand you back full reference gene, not just every gene on that organism, but all the enhancers, all the promoters of that gene. It gets even more interesting when you apply it to a human, because suddenly in a human, you can see all the structural variants. You want to understand why a cancer has certain variants and why it has translocations. But up to this point, our technology has not let us see the full sequence. So we have what are called driver mutations for cancer, and you can look at a little window of them. You can see maybe in some cancers 20% of the driver mutations. Our newest technology allows you to see all the way through. So that was a big moment, a big step forward. I got asked by Harvard to be on their Discovery Council, and they had one of the most preeminent genetics departments, and they had 24 professors. They wanted me to mentor one of them. And as you can imagine, this was a hard set of choices. I did a speed date called 24. I had gotten very good at interviewing geneticists by this point. And I said, what are you working on? Why is it important? And professor number 23 was a soft-spoken Australian, at that time unheard of, named David Sinclair. And he said, well, he says, I think I've come across a new class of compound that at least in animals seems to reverse the aging process. I said, good, I'm in. It got more fun from there. And so in 2013, we built Metro Biotech, which is focused really on the NAD space. And this is a fascinating space. You know, what is NAD? You know, you can see it at every drugstore now. But at that point in time, it was pretty much unheard of. We started building the precursors, those things that were small enough to go into the mitochondria and open up like flowers inside of the mitochondria as NAD. And NAD, as most of you know, I'm sure, is what allows your food to become energy. It allows the Krebs cycle to be active. And it's one of the things that are just so critically important in the conditions and diseases of aging. We have gone well beyond now. By 2016, we had created the first crystalline moiety of NMN. And this was an incredibly good precursor. It went into human blood, it went into the organs of the body profoundly. And this is now a published study. Within 2 weeks, we can raise NAD levels in a human by over 200%. And that made such an incredible impact in terms of the other conditions of the body. So these have now gone on, we'll talk about these toward the end of this slide, into late phase human trials. And we have a couple of really important phase 2s that have just been completed with really startling results. These are both in editorial review right now and will be published soon in a major medical journal. So you'll all be able to see them. But let's jump to what happened in terms of our own Edison lab. I don't know how to advance this. That's pretty cool. Did I do that? This is fine. So these are the major companies out of EdenRoc. Each one of them is pretty exciting. The top one is the one that everyone wants to talk about, which are the drugs that raise the NAD within a body. The second one down is also very neat. This is a cosmetic company that we built with NASA. And it actually came from our work in genetics. We were hired by NASA to validate that the Mars rover didn't contain any life forms, since we were so good at sequencing. They didn't want the rover to get to Mars and then think they had found life. So we were swabbing it down, and we found this very weird bacteria that had characteristics we had never seen in a bacteria, carried these sirtuin genes that are only found in higher life forms. And we said, wait, what's going on here? Now, our task was we'd take a peroxide and we'd take UV light and we'd swab everything down and try to kill everything. But this bacteria we couldn't kill. And we said, NASA, you know, what is this? This is in a great big JPL facility out in Los Angeles. And they said, oh, that. They said, we can't get rid of it either. Been here, now years. And we said, well, they said, would you mind sequencing it? They said, sure, sequence it, very strange genome. And everyone started debating, well, where'd this come from? And they said, well, all this stuff in this facility came from outer space. These are all the satellites that have returned. And so it was decided, put this thing in a little capsule, go send it up into outer space. And it went up to the International Space Station, sat on the outside of the International Space Station for a year and a half. We brought it back down, we opened it up, and to everyone's amazement, this thing was thriving. Oh, this is crazy. They said, no, this was during one of the periods where NASA was very depleted of funds. And they said, would you be interested in licensing? I said, sure. So we initially thought, oh, this would be a great sunscreen, right? It protects you from any UV light. Anyhow, it turned into a cosmetic. And this is one of them. It's actually a line of like 6 or 7 of them. You can buy it on Amazon, you can buy it direct. It's called Aeonia from Life, and made by the company Daily Science. You'll love it. Gets rid of wrinkles, does all kinds of things to your skin that are anti-aging. The next company down is Dovetail, which we spoke about briefly. Dovetail is really 2 separate companies. One of them is ArcBio. ArcBio is pretty neat. ArcBio is really a big problem that we tried to solve, that we could have never done with venture capital. With venture capital, if you know, says, hey, you guys, you got 5 years, we want our money back. So don't tackle anything that's bigger than a 3-year problem. Ours was an 8-year problem. So the question became, okay, we can sequence all life forms. I asked the question of our great scientists, how many things are there that can make a human sick to die? I mean, we're talking about viruses, bacteria, parasites, fungal. Anybody have an idea? This isn't all the strain, just the variant stuff, like COVID would be one. You assume millions, right? It's not. 1,400. That's it. 1,400 different organisms make you sick to die. So I said, okay, sequence them. Let's get their address. Let's get what they look like so that we can identify them. And they got through that. That took about 2 years. And then we had them in a soup. So we took someone's blood, and now we want to say, okay, do you find any of these things in there? Are any of these things present? And the answer was, 99% of it is human. We've got to somehow sort through all this and find the things that would make you sick, because this will take us all night and all day. We'll never get through it. So we spent the next 2 years figuring out how to cut up the human, which was a pretty interesting trick. And all that's left then are things that are not mammal. So now suddenly we could do it, but it took a mainframe computer to sort through the information. It took another 2 years to get it down to laptop size. It has been used for the last year at St. Jude's Children's Hospital for kids with cancer, and it's incredibly effective. And in 24 hours, they can tell exactly what these children who have cancer, whose immune system's already compromised, have, what they're infected. That's pretty neat, right? Talk about saving lives. Next company, Claret. Claret is a crazy company. Claret grew out of and really built the field of cell-free DNA. So DNA that your body has discarded, the cells of DNA that are discarded, are diced up. And when they dice them up, they're left in little pieces, 30 pieces to 100 pieces. For a long time, we thought that urine was sterile. You drink urine if you're stuck on a raft in the middle of the ocean. And then slowly we realized, no, there's things in that urine. There's little pieces of cell-free DNA that are sitting in that urine. But the pieces were too small to make sense of. On the other hand, cell-free DNA is not blunt on its ends. They have overhangs. They have overhangs 10 letters long. But nobody had ever looked at it because all sequencing technology start off with first step is blunt the ends. So they got rid of all the information, so there's very little information left. We built the 400,000 composite opposites that could mate with these strands of DNA. And suddenly you had twice as much information, and you could sort out what this meant. So we started using it for cancer detection incredibly effectively. 95% accuracy for prostate cancer without having to do a biopsy, PSA test. 97% accurate for liver cancer. But the most exciting thing that came out of it was that we realized that suddenly we could take extremely small pieces of DNA and identify them, and identify who they belonged to. And that grew into this company. So Astrea is now the leading forensics company in the world for degraded DNA. And the technology is so perfect that it has identified, well, it's done over 300 criminal cases. They're just constantly happening because people that have thought a case was cold can suddenly, if they have a single hair, a hair 1 centimeter long, identify the person in the world. And you're constantly shedding. So they did the Golden State Killer, which California, you know, was a big deal. But most recently, they did the Long Island. And this was a horrible kill. So a criminal who had killed at least 8 women. There's theories that maybe there's 26 others that are responsible. And nobody could figure out who. No. They gave us the DNA. We had some hairs left over. You know, it's a horrible criminal case. He would wrap the bodies in plastic, but the hair got into the plastic that he would wrap, and we identified it was him. But up to this point, we could only use this evidence in a circumstantial manner. They had to then go prove that he had a cell phone, calling these people. He was in this location. They had no other evidence. All they had was our technology. So they stopped the trial. They set about doing a Frye hearing, F-R-Y-E. And a Frye hearing is a fascinating hearing. A Frye hearing is where you prove a technology so far beyond a doubt that it alone can be used to implicate a person. It took 4 months. The jury approved it unanimously. And it became as good as the last time a Frye hearing was used was the fingerprints. So it's pretty neat. The Long Island killer pleaded not guilty. Pleaded guilty based on the evidence in front of him. He was sentenced to 4 life terms. He will never see light, David. Incredible. All of this came from trying to solve a cancer. Is that wild? So we'll flip to another slide since everyone just wants to talk about NAD and longevity. But before we get there, this is our leadership team. This is who created what we have in front of us. There's 62 world-class scientists. Many of these came from my tour to go meet the top 10. But you'll recognize many of the people, like Siddhartha Mukherjee, I'm sure you know of him, authored, Pulitzer Prize-winning book on cancer, leaders in cancer. Maybe you know Richard Durbin from Cambridge, the first real scientist of bioinformatics. Carlos at Stanford, who took it forward. Johan Ulrichs at APFL, probably leading person in mitochondrial disease, followed by Nick Blaine over here at University College London, who, as you know, is a prolific author, maybe 7 books on mitochondria. These are all what created what we have. The color rings that you see around them are the groups that they're affiliated with. So quickly, we have run out of time, but you can jump to the next slide. Next slide, these are all the people that work on EdenRoc. It's a team of 140. Next slide. Petro Biotech, what everyone wants to talk about. Next slide. Why does NAD work? NAD is no longer a question. NAD is in all your bodies constantly. How do you make it? You can't get it from an infusion. Sorry. You have to use a precursor to get in. Next slide. It doesn't just activate food pathways, it also activates the sirtuins that are critical for repairing DNA. Next slide. These are all the trials we've now completed. As you notice, we're deeply into human clinical trials. We're only maybe 1, 2, 3 years away from a registered trial. So this is perhaps the fruit of longevity right here. We know lots of things about it now. We've done metabolism trials. We've done all kinds of work. Most of this work is unpublished, but we have published 6 papers. So there's still a fair amount to be known. The 2 phase 2 trials are about to be published. One of them was in muscle strength and endurance. It's a remarkable study. We had already done a paper and study that's published on pre-diabetics, where we increased muscle strength without exercise. This muscle strength trial was in elite athletes. To get into that trial, you had to be able to run a 6.5-minute mile. Anyone in the room that can do that? There we go. You have to do it for 12 miles in a row to get into the trial. So these are all triathletes, all marathon runners. And just an exciting moment for you. It increased their endurance by 24%. You think about how crazy. It's confidential. We also did a trial on Alzheimer's that just completed 3 weeks ago. And in Alzheimer's, we found profound implications. The biggest one was crossing blood-brain. More to come from that. I don't know if you can switch to the last slide, and I'll sign off here. Let's skip all these. Go to the last slide. There we are. What? Oh, there we are. This is our current pathway, our roadmap. Everything in blue is a phase 2 clinical trial with humans, some of them very large, some of them very long. The green ones are phase 3, where we enter into late stage. So this is where we're at. I appreciate all your interest in longevity, and I think one day we're going to get there.